DNA is made up of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. There are 2 different types, purines and pyrimidines. The purines, adenine, guanine, are bigger, more complex with 2 carbon-nitrogen rings. The pyrimidines, T and C, and uracil and RNA have only 1 nitrogen ring. Purines always bond with pyrimidines. This is why A and T, or A and U in RNA, and C and G always bond.
DNA, deoxyribonucleic acid, holds the information for each organism's genetic material. It has a double helix structure made up of a backbone of phosphate and deoxyribose sugar. Attached to this backbone are the nitrogenous bases adenine, thymine, guanine, and cytosine. When DNA needs to replicate, an enzyme called helicase unzips the DNA, making way for another enzyme, DNA polymerase, which adds complementary DNA nucleotides. This process creates 2 identical DNA strands. The process is semi-conservative because the new DNA is made up of 1 original strand and 1 new one. RNA is made up of ribose instead of deoxyribose and uracil instead of thymine. In transcription, mRNA makes a complementary template of DNA to carry to the ribosome. DNA can't directly leave the nucleus because it is too large. RNA polymerase unzips DNA and produces a chain of complementary mRNA from RNA nucleotides. The mRNA leaves the nucleus and goes to the ribosome. In translation, mRNA is used as a template to make proteins. mRNA attaches to the ribosome at the start codon AUG. tRNA with the complementary anticodon and the corresponding amino acids come to the ribosome. When the next codon is read, the first tRNA moves aside for the incoming tRNA, leaving behind its amino acid. This process continues until the stop codon UAG is read. Then the amino acids break off of the ribosome and fold into a 3D protein structure which will come together with other proteins to form a trait.